Good morning, everybody, and thank Wolfgang for the nice presentation. Briefly, I would like to start my presentation with some statements. We know that the obese people shown a different metabolic and physiological configuration respected to the normal weight people, and we know also that they are characterized by a lower biodiversity respected to the normal weight people. And it has been suggested that these altered, this biotic profile of the microbiota can play a role in this rupture of the symbiotic relationship between microbiota and the host, so supporting a compromised host energy balance and the host immune homeostasis. So summarizing, we know that obese people show a low biodiversity and increased in the efficiency in extracting energy from diet, and an increase in some specific microbial groups which can sustain inflammation and insulin resistance, which are related to the obesity. But still an open question is the possible role of the microbiome in the onset of the obesity. And what we have done within the MyNewGut project, it has been to exploit two large cohorts of obese subjects, in particular children and women, who were recruited in previous European projects. And concerning the large prospective cohort of IDEFIX and E-family, they collected at a baseline more than 16,000 children. They did the first follow-up after 1 year, and for us this is time 1. And the second follow-up was done after further 4 years, and this is for us the third follow-up. In this context, in these two projects, they collected also feces, but only at time 1, during the first follow-up, and at the time 2. And as you can see, they collected the feces from a limited number of subjects. When we analyzed the data set, we could see that only 97 children had fecal sample collected at both the 2 time points. And for us, the more interesting subjects were these 70 normal weight children, because after 4 years, 36 were obese and 34 remained normal weight. Of course, for all these children, several metadata related to anthropometric lifestyle and, of course, also inflammatory profile, as well as other health parameters, were collected and used later on for the analysis. The 16S ribosomal DNA analysis of all these subjects showed a huge variation in the major component of the gut microbiome in both the normal weight and obese children. And we could find only small differences in the relative abundance of a very limited number of microbial groups, and in particular, proteobacteria and Bacteroidetes showed an increased relative abundance in obese children, whereas Christensen and Rache and sorry, I can't see anymore. Provetalace were increased in normal weight children. Then we analyzed by a clustering approach all the sequencing, and we were able to identify 4 major signatures of the gut microbiota profile, and in particular C1 and C2 were characterized by a very low abundance, a very low biodiversity, and 70% of the obese children showed this type of microbiota. The C3 and C4 signature of the gut microbiota were characterized by a very low biodiversity, and 70% of the overweight obese children showed this peculiar microbiota profile. And of course, we could also analyze which were the most abundant microbial group present in the normal weight children and in the obese children. Since we had a large panel of metadata and of parameters related to the health status of the kids, this is just a flavor of how many they were. We adjusted our model for age, gender, and the Tanner classification of maturation stages. And so what we could find, we could identify some significant correlation between the gut microbiota profile of these children and some of the major inflammatory biomarkers and health parameters. In particular, when we look at all the children at the 2 time points, we could see that there was a positive correlation between the C3 and C4 profiling of the gut microbiota with the increase of CRP, interleukin 6, I mean some immune markers, and other health parameters, including, for example, blood pressure. And when we look at the obese cohort at both the time points, so I mean also when the obese children were normal weight, we could find the same results. So C3 and C4 was strictly related with the increase of a huge number of inflammatory biomarkers. Then we look at the food frequency questionnaires, and we tried to find a correlation, significant correlation between the food consumed and the gut microbiota profile. And once again, we could see that C1 and C2 profile, the healthy profile, which was specific of the normal weight children, was related with the intake of some foods, including fermented milk. I have no time, I guess, to go into the details, but we could find other foods which positively correlated with the C3 and C4 profile of the gut microbiota, of course, including pizza, slide ham, sweetened drinks, and so on. So the conclusion, when we put it all together, we could claim that the combination of C3 and C4 microbiota profile with D2 and D5 diet, which means a diet particularly enriched in fat, in carbohydrate and with a very low content of fibers, is connected with the obese phenotype. Of course, what is interesting for us is that this combination was present also at the time one when the obese children were still normal weight. So we can suggest, we can speculate that the combination of a microbiome and the diet produce like a combination of factors which can be in some way predictive of a potential obesity, even if we know that it can only partially explain the etiology of the obesity. Then we performed, and these are unpublished data, when we perform a shotgun sequencing, analyzing only a smaller number of children representative of the two different microbiota profile and diet. And we could see that, and we could find that there, from the metabolic point of view, the normal weight children were enriched with genes involved in arginine and proline metabolism, and with genes involved in metabolic pathways involved in the short-chain fatty acid production. And on the other hand, the microbiome of obese children showed a increased relative abundance of some metabolic pathway, including fatty acid biosynthesis, unsaturated fatty acid, and histidine metabolism, and genes involved in the metabolism of starch and simple sugars. And what is interesting is that these differences were already present at T1 and were maintained at T3 in all the obese subjects. We performed on the same subject also a metatranscriptomic analysis, and once again we confirmed the data obtained by metagenomic analysis, and so an increase in the transcriptional level of genes involved in the production of short-chain fatty acid, and an increase in obese children of histidine metabolisms. And once again, these signatures were present also at the time one. So what we are doing now, we are trying to integrate in our model all these metatranscriptomic and metagenomic data, and we are also waiting for the metabolomic data coming from Professor Klaus. The second cohort I would like to introduce you is the neurofast cohort, which is a cohort of obese women addicted or not, food addicted or not food addicted. And one of our main goal was to identify the possible presence of distinctive patterns in the microbiome of addicted and non-addicted obese women. This is the design of the cohort. We analyzed the microbiome of 36 obese women with food addiction, 27 without food addiction, and 39 normal weight women. And the addiction was evaluated by a Yale Food Addiction Scale. I don't have time to go into the detail of this evaluation, and so I go straight to our results. We could demonstrate once again that by analyzing the 16S ribosomal DNA, that there was a huge variation in the gut microbiota profile of both the subject. But what was interesting was this data related to the alpha diversity because we found significant lower alpha biodiversity in obese women when compared to the normal weight women. And we could also identify some microbial groups which were more abundant in the normal weight women respected to the the obese women, and these include Ruminococcus, Coprococcus, and Roseburia. Then we tried to analyze the microbiota profile of obese addicted and not addicted women in order to find some possible different characteristic, different profile. And what we got, that of course this is just a preliminary data because the weakness of this study is the low number of subjects. It was that we could see that normal women without food addiction showed an increase in DORA, whereas Ruminococcus was the most abundant microbial group in obese women with food addiction. We performed once again the metagenomic analysis on a small representative number of women of the three groups, and what we can found it was a strong increase in the relative abundance involving metabolic pathway of sulfur, fatty acid, lipid, and carbohydrate only in women with food addiction. And the metatranscriptomic analysis suggested another significant difference in the transcriptional level of the tryptophan metabolic pathway, because as you can see, the obese women with food addiction showed a very low abundance of this transcriptional abundance of the genes involved in this metabolic pathway, which were increased in obese women without food addiction and was more increased in normal weight women. So, in conclusion, we can say that obesity is a quite complex mosaic of endogenous and exogenous determinants, and a microbiome can be considered only a single tile contributing to the obesity development. In children, we have seen that a low diverse dysbiotic microbiome configuration associated with an healthy diet and with an inflammatory status is characteristic of the pre-obese children, and that could be used as, eventually used as a predictive tool for analyzing the pre-obesity. And in obese women, we got the first very preliminary results about the possible role of microorganisms impacting on eating behavior, and so opening the way to the more detailed study about the role of Ruminococcus and Doria in food addiction. Finally, I would like to thank all the people who did this job, in particular the guys of my lab, in particular Simone Rampelli, the team from Bologna University led by Uberto Pagotto, and of course, Wolfgang and Catherine for the fantastic support in the study, and all of you for your kind attention.
Thank you. Questions? No questions? Please. Introduce yourself, please, and then.
Laura Fernandez from UFIC. Thank you very much for this interesting presentation, Patricia. Have you looked at physical activity patterns and the difference between...
Yeah.
And did you have any obese children that was heavily physically active? Or?
Yeah, we look at also at these parameters, and what we have seen that children with a unhealthy behavior, but with a high physical activity performance, they were normal weight in both. So,
thank you.
One more question.
Gareth Williams, Lancaster University. So, as I read the data, or the way you summarized the children's development, it wasn't necessarily an unhealthy microbiome that was causing the children to become more... it was the unhealthy diet pattern which probably was affecting the microbiome, which was then leading to the overweight, obesity.
That is a quite interesting question, but is egg or chicken? I don't know which was the first one. I mean, what we described was the situation in these children. So